Hi everyone, this is part two of talking about cellular respiration. We just have a couple of things to touch on here before we conclude the chapter. The first is that we're going to talk a little bit about other types of molecules that can be used by cells to make energy. And then lastly, we're going to finish up with anaerobic respiration, which is the process of making energy without oxygen. Okay, but first, we're turning attention here to the connections of carbohydrates, proteins, and lipids in metabolic pathways. So if you take a look at our 4 steps, remember we have glycolysis, which leads to pyruvate, and then pyruvate is oxidized to acetyl-CoA, and then acetyl-CoA goes into the citric acid cycle, and then that's going to be totally stripped of all the electrons and protons, and then those are used for oxidative phosphorylation. Well, the thing that usually glycolysis starts with is this molecule called glucose, right? But there's a couple of other different types of molecules, especially when we're talking about the carbohydrates. Not every carbohydrate is just single monomers or single sugars of glucose. You could have something like starch, which is a big long chain of glucose molecules, and that could then be converted to all of the individual glucose molecules, which can then be fed into glycolysis. Another example is that's pretty common is sucrose is a disaccharide, and this is made up of 2 monosaccharides. So what can happen is sucrose can be broken down into its individual monosaccharides, which are glucose and fructose. And both of these in turn then can be fed into glycolysis. Okay, so not just pure glucose is the thing that undergoes glycolysis. You have a bunch of different pathways that all sorts of a variety of carbohydrates can be initiating this whole metabolic pathway. And then above and beyond the carbohydrates, there are the lipids. And oftentimes, a couple molecules that get talked about when we're talking about lipids is this molecule called glycerol. Oops, I already misspelled this. And glycerol gets bound to fatty acids to form triglycerides. Triglycerides are kind of the main way that fats and oils store energy. So the fat that is on your body is mostly triglyceride, so that's made up of a glycerol with 3 fatty acids. Well, the glycerol can be converted into a substance that can undergo glycolysis. The fatty acids can actually be converted into acetyl-CoA, which is the final product of the pyruvate oxidation. So the fatty acids will then enter here and then undergo the citric acid cycle. Lastly, then we have the proteins. And proteins are a little bit different. Proteins are usually not used for making energy. They are made up of amino acids. And whereas these molecules here, carbohydrates and lipids, they're kind of one of their main purposes is for energy, but that's not so the proteins. Proteins have a variety of jobs. They go out and kind of do stuff. So they're not generally used to make energy for the organism. But in a scenario in which there are no carbohydrates or lipids, proteins can in fact be used to make energy. So imagine Professor Zach, am stranded on a desert island without any source of food of any kind. Well, kind of a grim picture. I hope it never happens to me. But if that was the case, my body would first burn up all of the glycogen. You know, glycogen is kind of our version of the animal version of starch. So there's a lot of glycogen in my liver, and there's a lot of glycogen in my muscles. Those would get burnt up to make, convert to glucose, and then that would be fed into this whole pathway to make energy. But then as soon as the carbohydrates were depleted, then the lipids would be burnt up, and all the subcutaneous fat, so all of the fat right underneath my skin, and in other parts of my body would be broken down, and the glycerol molecules would then undergo glycolysis. The fatty acids would enter the whole pathway here. And then if all of those were used up, then as a last resort, my body could use some of the proteins in there, and it could break those apart. Now, the amino acids can actually enter really any part of this whole metabolic pathway. There's 20 different amino acids out there, And depending on the amino acid, they have kind of a preferential entrance into this metabolic pathway. Most of them are able to enter right here into the citric acid cycle. So like if you take a look, here's our citric acid cycle. Pyruvate gets oxidized to acetyl-CoA, and then it goes around and around and gets totally broken apart. All of the electrons and protons stripped down. But you can have all of this stuff happening, all right? So these are all different types of amino acids, and you can see that there's a lot of different entrance points for these. Like, for example, all of these can get made into this thing, which can then get made into acetyl-CoA. You've got all of these amino acids, which can be made into glutamate, which could then enter here as ketoglutarate, and so on and so forth. And you certainly don't have to memorize all of these, but I just like to kind of show you the complexity here of how you can have all these amino acids being fed into this biochemical cycle. So that's a little bit about other pathways. One thing that's kind of interesting, when you do have amino acids that are being fed into this cycle, the first thing that has to be removed before they can enter is a NH3 group, or maybe it could be NH. I can't remember if it's NH3 or NH4, but we'll say NH3 for now. And if you take two NH3s and you add a little CO2 to the mix, it forms this product called urea. And urea is the thing that we urinate, right? That's why urine is called urine, because it contains a lot of urea. So, the nitrogenous waste, the wastes that have nitrogen in them that you urinate or get rid of out of your body, are going to be from kind of the breakdown of these amino acids. Now, generally, the stuff that makes up urea is just kind of the normal wear and tear of proteins. It's not necessarily a result of your body making energy out of those proteins. It's more like if the protein is old and it needs to be recycled, the amino acids will be broken down into urea. But that's kind of a little side note there, just of interest. You don't have to keep that in mind for the exam or anything. All right, lastly we're going to talk about then is the anaerobic cellular respiration. So anaerobic means without air, kind of literally in the Latin there, or in other words, without oxygen is kind of what we're talking about here. And one of the words that we generally associate with this types of energy is this word called fermentation. There's two different types of fermentation. There's a couple of different types actually, or a few different types, but the two big ones are lactic acid fermentation and then alcohol fermentation. The first one here, lactic acid fermentation, this is the one that's done by mammals and also bacteria. And a cool example of this is yogurt. So there's a bacteria out there called, well, there's a couple different types of bacteria that do this. I think it's called lactobacillus. And they are able to take glucose and break it down to form this stuff called lactic acid, and that's kind of one of the important parts of making yogurt. Well, here's a little bit of the process of what's going on here in lactic acid fermentation. Recall that generally, under glycolysis, glucose is going to get converted into 2-pyruvate. Now, in this process, some energy is made here. Okay, so this right here is the energy that can be used by the cell to do stuff. But recall also that in this process, when you have glucose being broken down to pyruvate, you have kind of a loss of electrons here. Because you're taking something that's bigger and you're breaking it down into something smaller. So you're kind of stripping off some electrons from that molecule. And the thing that picks them up are these molecules of NAD, like we talked about. So now you've got this molecule of NAD that's bearing 2 electrons. Well, generally, if there is oxygen, these electrons will get dumped over to oxidative phosphorylation. Okay, but this oxidative phosphorylation, this requires oxygen. Remember, this is kind of a good question too to ask. Why is it that oxygen is required in oxidative phosphorylation? And the answer is that as those electrons are passing along the electron transport chain, they have to go somewhere, and so oxygen is the final electron acceptor. It takes those electrons up so that the electrons aren't rattling, and in the process it makes water. Well, if there is no oxygen, then these electrons are not going to go to oxidative phosphorylation. So then the question is begged, how do we get back to this molecule so that we can keep this whole energy, this little smaller energy process happening here. Well, the answer then is that what happens is that this actually is going to dump its electrons off to pyruvate now, and in so doing, that makes this molecule back into the oxidized form, which can then feed back to be reduced again. Basically, in other words, to sum it up, this takes the electrons from glycolysis. The electrons have to be dumped somewhere before this molecule then can come back and receive some more electrons. Well, in so doing, what happens is that pyruvate is actually converted into lactate. Okay, so the big idea here is that in the presence of oxygen, glucose is converted to pyruvate, which is then converted to acetyl-CoA. Oops. Which is then turned into a lot of ATP through oxidative phosphorylation. Okay, that's when you have O2. When you have no oxygen, basically it's a shorter story. You have glucose, which is being converted to pyruvate, which is then getting converted into this thing called lactate. Now, lactate, oh, and by the way, this is going to make like 36 ATP, and in this process, you only really make 2 ATP. So it's quite a bit less efficient in terms of ATP production. But it can be done. All right, so lactate, what this does, we said that both mammals and bacteria are going to do this type of lactic acid fermentation. Sometimes bacteria will preferentially do this type of fermentation specifically because they do not have mitochondria. And this whole process of oxidative phosphorylation that happens when we have oxygen, this kind of requires mitochondria. So a lot of bacteria will undergo this type of fermentation. And then mammals generally prefer to do oxidative phosphorylation. So that's the one that requires some oxygen. But they can also, if oxygen is in short supply, they can kind of bypass the whole oxidative phosphorylation thing and just undergo this type of fermentation event. And in one example of this is if I was, let's say, running a marathon, or perhaps a better example would be, let's say I was in the gym and I was bench pressing some iron or doing some sit-ups or push-ups or something like that. What would happen is that as I'm exerting the energy on my muscles, they're going to burn up all of the oxygen, using it to try and make as much energy as possible. It takes a lot of ATP to make your muscles carry heavy loads, and if I'm working out my muscles pretty hard, they're going to burn up all that ATP. So initially there's going to be some oxygen that can supply my muscle cells with the required ingredients required to continue making ATP through oxidative phosphorylation. But there's probably going to come a time at which my energy demand overtakes the amount of oxygen that's available. And in that instance, suddenly my body's going to kind of have to start making some lactate because there isn't as much oxygen available to continue this whole process. So it's kind of this is kind of like cheap and easy to make energy when you do this type of process, because you make 2 ATP. It's made super fast. It doesn't require oxygen, but a byproduct is lactate. This lactate will build up, and what happens is that it kind of makes the whole muscle cell acidic, because this actually turns into lactic acid. So it makes your whole all of your muscles kind of acidic. And if they get too acidic, if I keep bench pressing that iron, or I keep doing push-ups or sit-ups or something, eventually there's going to be so much acidity in my muscles that some of the proteins are going to denature, and they simply aren't going to work as good as they ought. And many of us have probably experienced this at some point in time. And if you want to experience this, go into the gym and try and do a whole bunch of push-ups, okay? Because you will initially be okay, you'll be able to do those push-ups, but as you keep doing the push-ups, eventually there's going to come a point where you can hardly get your muscles to work anymore. You're just going to drop to the floor. And what's happening there is all of the lactate that's building up is making your muscles acidic, so that some of the proteins are stopping to work, or they're not working anymore. And so then you have to stop your exercise, and then some of this lactate will get cleared out, and then the acidity comes back to normal, and then after you'll probably have some sore muscles, but then they can work again. Okay, so that's lactic acid fermentation. The other type then is alcohol, and this is one that's done by yeast. And yeasts are a type of fungus, so they're not a plant, they're not an animal, they're a type of fungus, and they are single cellular fungi. And here's what they do. They take pyruvate and they convert it to pyruvic acid. And then you don't really have to keep in mind all of these steps, but the big thing is that what is made is this product called ethanol. And ethanol is the alcohol that's found in, like wine and beer and spirits. It's also the type of alcohol that's found in, like if you're baking leavened bread and you put yeast in the dough, and what it's going to do is kind of break down those sugars and produce ethanol and CO2 as a byproduct. Here's the CO2, here's the ethanol. And that is another type of fermentation. All right, so a little bit there. I've got one last review question here in this chapter, and then we'll be done with chapter 7. So A, in the absence of carbohydrates, how would an organism make energy? B, in the absence of oxygen, how do mammals and bacteria produce energy? And C, what is produced when yeast ferment sugar? Go ahead and pause the video and see if you can work on some of these. All right, so let's take a look. Part A. In the absence of carbohydrates, how would an organism make energy? So this is, again, we've got our 4 steps. We have glycolysis. We have pyruvate oxidation. We have the citric acid cycle. And lastly, we have oxidative phosphorylation. All right, so we said glucose is the thing that normally gets fed into glycolysis, but we could have starch be converted to glucose, and then that would go in. We can also have something called sucrose get converted into its individual monomers, which are glucose and fructose. And glucose can certainly go right on in. And I believe that fructose, can get converted to actually go in as well. I'm not sure if it gets converted to glucose or if it gets converted to something like glucose, but in either case, there's a couple examples of different types of carbs. And then remember the lipids, we had 2 molecules, glycerol and fatty acids. Glycerol undergoes glycolysis. Fatty acids get converted to acetyl-CoA, and they go in here. And then the the proteins are made up of amino acids, and they kind of go into every different part. But most of them probably go into the citric acid cycle. All right, that's that answer. In the absence of oxygen, how do mammals and bacteria produce energy? Well, the answer is fermentation. Specifically, if we're talking about mammals and bacteria, though, we would say that this is lactic acid fermentation. So basically, glucose, how this process works, glucose is converted to pyruvate, and then pyruvate is converted to lactate, and that will generate some ATP. Okay, so that's that process. And then lastly, what is produced when yeast ferments sugar? And the answer here is ethanol. Sometimes it's referred to as ethyl alcohol, which is a type of alcohol. All right, there we have it. Chapter 7 wrapped up. Good luck as you continue on your studies. Next up is Chapter 8, which is all about photosynthesis.